All right, good morning. This is Dr. Peterson, and this is my first online pre-lab lecture for computer-assisted molecular structure visualization. It's my first attempt at this, so bear with me. It's Tuesday, March 17th, about 9 a.m. in the morning. I haven't showered yet, don't judge. I'm sitting in my kitchen with my daughter. You want to say hi, Trixie?
Hello.
And we'll see how this goes. So our goals for this experiment are to gain experience working with the molecular modeling program and to learn about chemical structure and bonding. I thought I'd start with the first point. I often point out to students that this is the most important plot in chemistry, in my humble opinion. What it is a plot of the change in potential energy, and let's see if I can get my pen to work here. This is potential energy over here, as a function of the internuclear separation between two atoms. So imagine these guys here are atoms, and what you think about is how the potential energy changes as these two atoms get closer and closer together. This is the way we normally think about it. At large separation, we imagine that the atoms are so far apart that they can't interact. Right? And if they're so far apart they can't interact, their change in potential energy is zero. As they start to get closer and closer together at some intermediate separation, the electrons from one atom can interact with the nucleus of the other and vice versa. Right? This is a stabilization, right? The negative electron is attracted to the positive nucleus. It lowers the energy of the system and ultimately provides some stabilization to the molecule. If you keep pushing the nuclei closer and closer together, at some small separation, the repulsion between nuclei dominate and it becomes destabilized. Now, the reason this is such an important plot is because you get two critical pieces of information. Right? The first is this guy here, the bottom of this well. And if you think about it, what you might recognize is that this quantity right here is the bond distance. Right? That's supposed to be a T. There we go. That's how far the atoms are apart. This quantity here, the vertical version of this, is the bond energy. And so you can imagine that if we had a computer program that could simulate these things, we could get these really important molecular parameters, you know, the bond distance and the bond energy, without actually doing any experiments. In addition to bond lengths, bond distances, we can get some other geometric quantities. So this is the first one I just talked about, bond length. Bond length I would describe as a one-dimensional quantity, meaning that you could define it on a number line, and ultimately, in order to characterize a bond length, what you need are two atoms. Okay. The next one that we can get is a bond angle. A bond angle takes three atoms, this one, 2, 3. To define it, we'd have some angle alpha, and because this exists in a plane of the paper, I would say that it's a 2D quantity. One more, if there's a 1D quantity, a 2D quantity, you might imagine that there is also a three-dimensional quantity. And this is the hardest one to visualize. It's called a torsional angle. I tried to steal some picture from the interweb rather than draw it for you, but this is an ethane molecule, C2H6. Each of these black atoms here are carbon atoms. And what you could imagine doing is looking down the bond angle, right? I'm not going to try and draw an eye, it's not going to work out well. But imagine looking down this bond, and what you would think about is the twist between atoms on the back. This is a hydrogen atom on the back. This is a hydrogen atom on the front. And the twist between the atom on the front and the atom on the back is called the torsional angle. It will ultimately take 4 atoms to define. 1, 2, 3, 4. and is a simple three-dimensional analog of this bond angle. At the end of the day, all of these parameters, bond energies and molecular structure, are critical for determining the functionality and reactivity of a molecule. Some of you are biochemists, biology-oriented people, and you probably have heard of structure-activity relationships. The idea is the structure of a molecule or a protein or some biological entity determines its activity, its biological activity. And so modeling programs are important because they let us get at all of these quantities without doing any actual lab work, right? We can simulate and get some idea of what the answer might look like before we actually go do anything. No matter what any of you go on and do, you will go on and do some modeling programs in some capacity. And so in that sense, I think this is a really good lab for you to start to learn about how these things work and how we use them. Got to give you a little bit of biology background. So one of the molecules that you're going to look at in this lab are called amino acids. There are 21 naturally occurring amino acids, and these are the building blocks from which proteins are formed. Here are 4 of them that are shown: arginine, histidine, alanine, and lysine. And I want to point out to you some common factors in the structure of these molecules. Oh, my son Gus just walked downstairs. Gus, do you want to say hi really quick? To my class. Come over here and say hi. He just woke up. He has no school today. You say hi?
Can they hear me?
Yeah, they can hear
me?
No, they can hear you.
Oh, hello.
Okay, that's my son Gus. Anyways, here are the molecules. So each kink in here represents a carbon atom. There's a carbon atom here, there's a carbon atom here, and if you haven't seen these kind of structures before, you will shortly.
Where's Trixie and Mom?
Trixie's downstairs. If you look at these structures, you'll notice that they all have some common components, specifically this group here. Each molecule, you'll notice, has a NH2, OH, C double bond O, right? These things here. And there's something which is unique to each of these. So what's pegged onto the end of these is unique to each amino acid. This one's different than this one, right? They all have different things. I need to tell you some information about these, and I guess let's start over here. This group has really 3 different parts. So the first is called a carboxyl group. This C double bond OOH is called a carboxyl group. The nitrogen group here is called an amine or an amino group. And then sitting right in the middle of it is a carbon atom, which we call the alpha carbon. It's the first one. The other half of these things is what's known as a side chain. The side chain is what's unique to each amino acid. And it's important because the side chain is what determines the individual properties of the amino acids, right? They might be hydrophobic, they might be hydrophilic, they might be acidic, they might be basic, whatever. You're going to explore some different amino acids in this lab and learn something about their structures. So here are my specific instructions for today's lab. Step 0 is that you need to download this JMOL Chem119 folder to your computer. Instructions are on page 133 of the lab manual. I also may try to put them up on Canvas. That may be a little bit easier way to access, but you'll need to get this Chem119 folder onto your computer. I think the easiest way to work with JMOL is essentially to play with it. You can load molecules, you can learn how to rotate them, zoom them, translate. The specific formalism for figuring out how you measure things is a little unique. It's a command line interface. So if any of you are Linux people and you like working with command lines, this will be familiar to you. Follow the instructions in the lab manual and you'll get a hang for how you measure bond angles and bond distances and torsional angles, all of that kind of stuff. Once you get a feel for working with it, you'll want to measure the distances, angles, torsional angles as directed in tables 1 through 6. Right? It will have you start with some simple hydrocarbons, and then you'll get to some more complex inorganic molecules. You're going to measure things and then enter them in these tables 1 through 6 as directed in the lab manual. I would write the numbers right into your lab manual, and you're going to use them to calculate some things later. After you do the inorganic molecules, you're going to measure amino acids, and this is the first important thing I need to tell you. When you measure geometric quantities on the amino acids, only measure the side chain. Oh, that's the worst. I'm still learning how to use my pen. But only measure your side chain. Right? You'll want to make sure that you note the number of bonded atoms and whether this carbon is in a linear or ring arrangement. The reason is that this is information that you're going to use later in the analysis of the lab. The other thing I'd point out to you is that you don't necessarily have to fill in all of the columns in tables, for instance, table 4, for the amino acids. Right? There's more slots available than you'll need to fill. At the end of the day, what the report sheet is going to ask you for is an average bond length for a carbon with 3 bonding partners. Right? You don't necessarily need all the data. What you need is enough data to get an accurate average. If you have questions, let me know, but that's my advice to you. No, Trixie, you may not draw something. After the amino acids, what you're going to do is look at some protein structures. And these are really cool. They're from published protein structures that are stored in something called the Protein Database, PDB. There are instructions in the lab manual for how you load those. It's pretty simple. Each protein has a specific molecule name. You're going to load the protein and then use the command line interface to adjust the way you visualize and see that molecule. Again, my advice is to play around with it until you get some feel for it. And again, at the end of the day, what you're going to measure here is some geometric factors like distances between iron atoms or a zinc nitrogen distance. If you try to follow the procedures that's written in the lab manual, it probably won't work really well. Instead, what I would advise you to do is to become familiar enough with the program that you can use a procedure that works well for you. Right? All right. Last but not least, in terms of submission, what we're asking you to do in this lab is to download the .docx file from Canvas. This is the Word file. Look at your data as you've written it on your lab report, calculate the quantities that it's asking you to calculate, and enter them into this docx file. Once you have all the data entered, right, you will submit it via the Canvas Dropbox. The assignment is due Friday, April 17th at 11:59 p.m. And this is all sort of new to us, so if you have any questions or concerns, please contact me as soon as possible. I'm happy to help you in any way that I can. So, my notes say this was 13 minutes. I hope that was an acceptable time. My daughter wants to draw you a smiley face on my pad, so she's going to do that now. Let's see how it goes. Oh, draw it down here.
Oh.
Yeah, somehow it ended up looking more like a frowny face, Trixie. Here, let's see if I can do a better one. Hang on. Oh, she wants another chance. Well, Trixie, let's see. There's one. All right, there's my smiley face. I hope you're all doing well. Have fun, it's a good lab, and I will be here to help if you need it. All right, bye-bye.